We're going to look at chapter 27 today. This will be our last lecture, our last meeting. So anyway, let's make it a good one. Overview of human development. Okay. Okay, human development. We're talking about all those factors that involve pregnancy and development. Developmental biology refers to the science that studies changes in form and function from a fertilized egg all the way through to old age. You know, it's basically our existence here on planet Earth. You know, embryology specifically is a study of all those events that occur during approximately a 38-week period of gestation, or that period of development inside the mother's womb. Gestation is basically from the last menstrual period to delivery, okay? So that's what we refer to as embryology, okay? So the prenatal period is everything that happens in that 38 weeks within a mother's womb before birth, okay? That period is sometimes referred to as a pregnancy period. So, but as healthcare professionals, we call it the prenatal period. Okay, the postnatal period is all those changes that occur in life after birth. Natal refers to birth. Okay, so of course our environment with an atmosphere, with gravity, that's very different for the baby once it's born compared to its previous environment inside the embryo, inside the womb, where it's in a fluid environment. Totally different type of environment. So the physiological anatomy and the structures have to adapt to, you know, existence, you know, with gravity and an oxidizing atmosphere. Okay, so the postnatal period will be very different from the prenatal period. So there's 3 stages of prenatal development that occur during a 38-week time period. And those include the pre-embryonic period. This basically lasts for the first 2 weeks immediately after fertilization. The zygote, remember, a zygote is a fertilized egg, okay? That divides mitotically. Remember, that's not meiosis. Mitotically, just a reproduction of cells many times to produce a multicellular structure called a blastocyst. It's the blastocyst that implants in the endometrial wall, okay? And that implants in the endometrium of the uterus. Okay, so even though cleavage occurred in the oviduct right after fertilization, cleavage just means the division by mitosis with this exponential growth. Like, so 2 to 3 days past fertilization, the cleavage produces what we refer to as a hollow sphere of cells called a morula. And in the morula, this is going to become a blastodisc. And the blastodisc is what's going to implant in the endometrium. So this is what implants in the endometrial wall. So we refer to as the embryonic period, that period between weeks 3 through 8 of gestation, that very early period of life. Conceptus is another term for zygote. The conceptus will develop and grow, it folds, it forms these rudimentary organ systems, and that's what we collectively refer to as an embryo. And so embryonic development or embryonic period is basically alluding to the embryo. And then the final stage of the pregnancy or the gestation is what we refer to as the fetal period. This is when the fetus looks more human-like. Okay, it lasts from weeks 9 through 38 of gestation. A normal period of gestation is about 38 weeks, or until birth. The conceptus is what we refer to now as a fetus. It grows larger and continues to develop until all the organ systems can function without any assistance from the mother. Okay, that includes, you know, the GI system, the urinary system, everything. Okay. And then after birth, the fetus is what we refer to as a newborn. And then that's when the postnatal period begins. Okay, so prenatal and postnatal. So a neonate just means an early newborn, okay? Like when you work in a neonatal intensive care unit, That's when they get these little babies that are born that are in critical need of attention. Okay, and that's when they take the top nurses and healthcare professionals to get into that field. Okay, because it's a very critical field. So anyway, this is a diagram that shows you the summary of the developmental changes that occur during this process of prenatal development. As you can see, the blastocyst is what implants inside the endometrium. Okay, remember this is the result of cleavage. Okay, so the zygote divides mitotically many times to produce a multicellular blastocyst that then implants in the uterus. Okay, so that's weeks 1 and 2 post-fertilization. Weeks 3 through 8, that's what we refer to as the embryonic period. The blastocyst will grow. As you can see, it's looking more like a human, and it forms the rudimentary organ systems. All of the rudimentary organ systems are basically laid down by week 8. The conceptus is now what we refer to as an embryo. And then finally, the fetal period is the weeks 9 through 38 until birth. Okay. Now the conceptus is now called a fetus. It grows larger and develops until the organ systems can now function independently. Okay. So those are your 3 periods. Okay. So the postnatal period extends from birth to 1 month of age, and infancy then will continue development through about 2 years of age. So when we refer to infancy, we refer to that time period between 0 and 2 until it becomes a toddler. Okay, childhood will then last until the period of sexual maturation or puberty, and that of course is when the brain starts producing hormones. At that point, adolescence starts, and that means the individual is capable of sexual reproduction. Adulthood then will extend from the end of adolescence all the way until death. We have early adulthood, young adulthood, middle age, and then old elderly. Okay, so the stage at which development ends and degeneration of the tissues and organs is sometimes referred to as senescence, and that's how we get the term senility. Okay, eventually senescence will lead to death, even without any specific disease state. Human beings, don't really live with a very good quality of life, past, when you get towards 80 and 90, okay? So let's look at the pre-embryonic period, fertilization through implantation. So the first thing that happens is fertilization, okay? That pre-embryonic period begins with fertilization, the sperm cell uniting with an egg to form a zygote. Then the blastocyst moves into the uterus, implants in the endometrial of the uterine wall. Some cells of the blastocyst begin to form what we call the extra embryonic membranes, which help support the developing embryo. These extra embryonic membranes are going to support the placenta, which is going to provide nutrition. Meiosis is what produces the gametes, the sex cells, the sperm and the eggs. So we have genetically unique haploid cells, which are 1N. Remember, they have half the chromosome number normally found in your body cells, which contain the diploid number. And then when the zygote is formed, then the diploid number is restored. So both gametes contribute equally to the chromosome number, and therefore to the genetic makeup of the new offspring. Sperm cells, remember, they have to travel a very long distance in a fairly formidable environment, being the vaginal tract. So they need to be small and motile. Okay, so they don't really need a lot of cytoplasm. Their main function is to provide the DNA, the nuclear material. Whereas the secondary oocyte is relatively large. It's almost 100 times larger than the sperm. And that's because it provides all of the cellular machinery, the nutrients, and basically the sustenance to maintain that first week of development. The cilia lining the uterine tubes and those gentle peristaltic contractions of the oviduct will help repel the oocyte toward the uterus. So the oocyte is really not motile at all. It depends on those currents that are created by those ciliated epithelial cells that line the oviduct. Whereas the sperm are extremely motile, okay, by that stage. So first, the sperm cells are ejaculated in the semen. Remember, ejaculated means they basically are moving through the ducts. So you have a mixture of sperm and fluid secretions from the accessory glands like the seminal vesicles and the prostate and the bulbourethral gland. And those are then mixed with the sperm, which is less than 5% of the total mixture. Okay. The ovulated secondary oocyte is only viable for about 24 hours after it's released from the ovary. And it's usually found in the distal end of the oviduct, where the ampulla, that widened area. That's usually where you find the fertilization occurring. And again, that is a test question. Okay. Where does fertilization occur? In the ampulla of the oviduct. Pregnancy most frequently occurs when the sperm enters the female reproductive tract during this 3-day window. And usually, you know, when couples are trying to conceive, and if they've had difficulty, a lot of times they'll have a thermometer and they'll do make sure that the mother is ovulating. And they'll, usually when the mother is ovulating, or the potential mother, she generally has a 1 degree centigrade higher temperature, body temperature. And so they usually practice coitus during that window period of time, mainly to increase the chances of producing a baby. Anyway, the pregnancy most frequently occurs when the sperm enters the female reproductive tract during this 3-day window between days 13 and 15. And at that time, of course, the female, the temperature will be a little bit higher, and also the breast will be a little bit reddened. Okay. And so either from 2 days before ovulation to 1 day after, that's the window period. 2 days before, so between days 12 and 14, and then day 15. So 12 through 15, that's the window period of time. But sperm can actually remain viable in the female reproductive tract for up to 5 days. Okay. But ideally, it's between those 3 days, 2 days before and 1 day after ovulation. So before it can fertilize the oocyte, remember, sperm have to undergo through a process called capacitation. And these are basically functional changes in the sperm that make the sperm fully motile. And also they modify the plasma membrane of the sperm so it can actually fuse with the secondary oocyte plasma membrane. So the sperm move kind of like with a whip-like motion, kind of a corkscrew fashion, with their flagella, their tails. And there are also numerous prostaglandins secreted by the prostate gland in the semen, and these stimulate those uterine contractions, which assist with the movement of the sperm farther into the female reproductive tract. Even though the female orgasm is not required for fertilization, it does help in moving the sperm through the tract, through the oviduct. So this process whereby these sperm are moving up into the female reproductive tract is called sperm migration. And then there's a very important reaction called the acrosomal reaction. Without this reaction, the sperm cannot inject the nuclear material into the egg. Okay, so the ovulated secondary oocyte is surrounded by a thick layer of granulosa cells called the corona radiata. Corona refers to crown, okay? Like the coronavirus, you know, as we all have heard. Okay, you probably already heard this before. The corona refers to crown, so the corona radiata is basically that group of cells that surround the secondary oocyte. Okay, they're actually follicular cells that were formed during the process of the vesicular follicle production. So anyway, the sperm must penetrate this corona radiata by a combination of both sperm movements and an enzyme called hyaluronidase. It's a very powerful enzyme, but you need a lot of it, okay? This enzyme is released from the acrosome, which is on the top of the sperm head, on the head of the sperm, okay? Hyaluronidase is the enzyme in that acrosomal cap. So if you look at the sperm, you've got your, it looks kind of like this, and the head, and then you have the nuclear materials at the bottom, okay? And then the acrosome is at the superior end, okay, in here. And then you got the mid piece comes down with a lot of mitochondria, and then you have the flagellum, okay, which is the whip or the tail of the flagellum. Okay, so this is kind of what a sperm looks like after it's fully formed. Okay, and of course the sperm are basically just microtubules that are connected by these arms that move past each other. That's what gives the sperm its motility. Okay, but remember there's between 40 million to 750 million of these. Okay, so the acrosome enzyme hyaluronidase is located here, okay, at the very top of the nuclear material. Okay, this is the nuclear material down here. Okay, this is the nuclear material, the DNA right down here. Okay, so that's probably the second most important function of the sperm. Of course, most important function is provide the DNA, of course. Okay, so that it can produce an individual. But the second most important thing is basically to have that acrosomal enzyme, that hyaluronidase, to break down the cell membrane so the nucleus, the DNA, can get inside the secondary oocyte. Hyaluronidase will catalyze reaction that then break down this corona radiata, the protein layer that serves as the barrier between the granulosa cells in the corona radiata. Right deep, just deep to the corona radiata is a very tough cell membrane called the zona pellucida. It's a tough layer of extracellular matrix created by the oocyte, and what it contains are these sperm binding receptors. And these, when the sperm cell binds to the sperm binding receptor, the calcium channels will present in the sperm plasma membrane. They open up, and that allows the calcium ions to flow then into the cell. So that increased intracellular calcium will then trigger the acrosomal reaction. Okay, and by the way, this is another test question. This is another test question. Okay. It triggers the acrosomal reaction. So there might be a question that asks, what triggers the acrosomal reaction? It's basically the influx of calcium. That releases the acrosomal enzymes, which contains additional hyaluronidase and acrosin, and also it helps digest those little holes in the zona pellucida. There's also several hundred sperm cells that are needed to release enough of this enzyme, this hyaluronidase and acrosin enzyme, in order to break through this corona radiata and also the zona pellucida, which is equivalent to the cell membrane of the egg. So when the first sperm penetrates the zona pellucida and then binds to the oocyte's plasma membrane, there are immediate changes in both the oocyte and the zona pellucida, so that no more sperm can now enter. Okay? So you don't get polyspermy. If you had polyspermy, of course, you'd have more than one copy of nuclear material. Of course, that would produce a miscarriage. So the sperm entry will stimulate the cortical reaction, and it destroys the sperm binding receptors. Those specialized secretory vesicles called cortical granules in the oocyte contain enzymes. They then catalyze all those reactions that destroy those sperm binding receptors. And the sperm entry then triggers a cortical reaction, which causes the release of enzymes from the cortical granules. Okay, so therefore, any additional sperm cells are rejected because of these cortical granules. This cortical reaction prevents any additional sperm cells from binding. On rare occasions, though, there are more than one sperm cell that enters the secondary oocyte, and that's called polyspermy. And when that occurs, development cannot proceed any further because the fertilized oocyte is now triploid. It's got 3 copies, and therefore that's going to be fatal to the embryo, and there will be a miscarriage. Okay. So what happens immediately is the plasma membrane of the sperm will bind with the plasma membrane of the oocyte, and 2 membranes fuse. The cytoplasmic contents of the sperm cell, including the nucleus, then are released into the cytoplasm of the oocyte. So with the sperm cell nucleus swelling, enlarging, that's what we refer to as a male pronucleus. Okay, so the oocyte will now be triggered to complete meiosis 2. Remember I said meiosis 2 is suspended in metaphase 2 unless fertilization occurs. Okay, and this is the fertilization event. And the resulting ovum also will swell to form a female pronucleus. Okay, so you get these female and male pronuclei that swell before they unite. Okay, at the same time, the plasma membranes will signal the secondary oocyte to complete meiosis II and then become an ovum that can be fertilized. So when the other secondary oocyte that resulted after meiosis, it becomes a polar body. Okay, that's then expelled. Sometimes it remains visible at just the edge of the ovum. So that's what we refer to as a female pronucleus, as a secondary oocyte nucleus swells. Also, you have spindle fibers between the male and female pronuclei. It looks very similar to what happens in the second stage of meiosis, in meiosis II, during the anaphase. The pronuclear membranes will break down, allowing the chromosomes then to mingle. But if any part of the process is defective or interrupted, you won't have reproduction. Okay? Infertility means the inability to produce a pregnancy after 12 months, 1 year of unprotected sexual intercourse. So anyway, that's the process of fertilization. Okay. As you see from this little diagram here. Okay. So here's the sperm undergoing capacitation. Remember, capacitation occurs before fertilization. Okay, and then they migrate to the oocyte, the acrosomal reaction. Then that basically occurs and allows the enzymes from the head of the sperm, and now the sperm will be able to bind to the plasma membrane of the oocyte. Then you get sperm entry. And that basically initiates the cortical reaction, which destroys the sperm binding receptors. And this will prevent polyspermy. And eventually additional sperm cells are now rejected because of the cortical reaction occurring. This is that zona pellucida I was referring to. Okay, some people call it the secondary oocyte cell membrane, but it's much stronger and thicker than a cell membrane. So it's a modified cell membrane. Here's a polar body that resulted from the completion of meiosis II. Okay, so here's the sperm nucleus, and it's going to form a male pronucleus. And here's the oocyte nucleus completing meiosis II. Okay, and then it forms a female pronucleus. And then finally, the spindle forms between the two cells, and the female pronuclei and the male pronuclei unite, and the chromosomes basically intermix, as you can see from the diagram. So that's what happens in fertilization. And there you go. That's going to have the beginnings of a whole new individual. Okay. So cleavage, approximately 30 hours after fertilization, the zygote undergoes cleavage. That's very rapid, and you get this exponential increase in genetically identical cells. Those are referred to as blastomeres. Divisions occur so rapidly, though, the cell number increases, but without any increase in size of the cells. Okay, the cell size actually becomes progressively smaller. Okay, so each subsequent division will take less time than the previous one, and by the second day, it goes from 2 to 4, then it goes to 16 cells, and you get this exponential increase, okay, of cells. And that's what we refer to eventually as a morula, a hollow sphere of cells. So the cells will then start to differentiate, and these are going to produce the building blocks of all future tissues of this developing conceptus. This is what we refer to as a morula, just a hollow sphere of cells, which contains the precursors for your germ layers. And it also still remains covered, though, by the zona pellucida. So by the time the blastocyst is ready for implantation in the uterus, in the endometrium, it has two distinct cell populations. The outer layer is what we refer to as flattened cells. These are your trophoblast cells. And these are what surround your fluid-filled cavity, and they also help participate in forming the placenta. The placenta is a temporary organ for providing nutrition for the developing fetus. Okay, all the nutrients and oxygen are going to be provided from the placenta to the conceptus. So the placenta will have the maternal circulation intermixing with these little structures called chorionic villi, and this is where the blood vessels between between the mother's maternal blood supply, where they're going to basically provide the oxygenated blood to the fetal circulation, which is going to, of course, contain deoxygenated blood in the, of course, the umbilical arteries. So it also helps suppress the mother's immune system, preventing it from attacking the conceptus, the placenta. The inner cluster of cells is called the inner cell mass, or the embryoblasts, and these are what form what we call the embryo proper, the developing body. That's what we refer to as the inner cell mass. The outer cell mass is the placenta. Inner cell mass is actually the embryo. So this is basically the cleavage. As you can see, cleavage occurs in the oviduct even before implantation. Okay. So fertilization occurs either between day 13 through 15. So after day 1, this is what you see with the zona pellucida over here and over here. This is day 1. Here's the male and female pronuclei. Fertilization has occurred and formation of pronuclei has occurred. Now you have, of course, it occurred in the ampulla, and now you've got a zygote. Okay, so that's about the stage, about day 1. This is about where that early zygote is going to be located. Okay, and you can see the spindle fiber starting to form right about here, midway down in the oviduct. Okay, day 2, now you got these blastomeres that form. Okay, the 2-cell stage then goes to a 4-cell stage, which then goes to a 16-cell stage, and then you get this hollow sphere of cells called a morula. Okay, and that eventually will become a blastocyst. Okay, where you get this infolding occurring, and it'll occur on day 4, just prior to implantation. So the blastocyst is now going to be hatched from the zona pellucida, okay, as you can see here. Okay, and then on day 4 to 7, you can see the inner cell mass and then the trophoblast. Okay, the trophoblast will be the placenta, and the inner cell mass will be the embryo. And that's going to be in this little area right here, implanted in the wall of the uterus and endometrial lining. So occasionally, within the first 8 days after fertilization, cells will separate into 2 groups, and they develop into 2 individuals. Those are referred to as monozygotic identical twins. That means literally single egg twins. Single egg, single sperm. Okay. And that's much more rare than fraternal non-identical twins. Fraternal twins is not a very uncommon thing to happen. But monozygotic twins only happens in less than 1% of the population. Okay? It's just kind of a freaky thing that happens where you've got clones of an individual. Okay? But they have nearly identical appearances. Okay? And they have the same DNA. And they both will share the same placenta. So very rarely you can have identical triplets or even quadruplets that result from separations during these early cell divisions. Remember, this is during the stage of the morula development. So, I mean, that's why it's so remarkable. Dizygotic or fraternal twins just means it's a much more common type of twinning. It results from ovulation of 2 secondary oocytes at the same time. So they're going to be fertilized by 2 separate sperm cells. Just what happens, the same thing that happens when a cat or an animal has a litter. Okay, the individuals may be of different sexes and very dissimilar in appearance. They're like siblings, but they're going to develop at the same time. Okay, the individuals may be different sexes and very dissimilar in appearance. Rarely you'll see multiple ovulations resulting in triplets, quadruplets, or even greater numbers of offspring, especially if they go to fertility clinics and they're placed on medications to increase their chances of having a successful pregnancy. And that's because the medications they take spawn, kind of precipitate this excess fertility period. According to CDC, in 2014, 1 in 8 couples in the U.S. had trouble getting pregnant or sustaining a pregnancy. In males, the most common cause of infertility is a low sperm count. In females, you have multiple causes. One is hormonal problems associated with ovulation. Others are diseases of the ovary, like polycystic ovarian disease, or fibroid tumors. Obstruction of the uterine tube is the most common reason for obstruction of the uterine tubes is from endometriosis. Endometriosis means abundant endometrial tissue in an organ other than the uterus. Okay, remember the endometrium should be in the uterus only, not in the oviduct. But when it is present in the oviduct, that will somehow be an incentive for the fertilization and the implantation. Okay, and that's the reason once you clear that endometrial tissue, you can avoid having all these miscarriages. Okay, and that's usually what happens. The mother will have miscarriages. She'll think she's not ever getting pregnant, but she's probably had several miscarriages because what happens is that endometrial tissue, the zygote thinks it's the endometrium, then it implants it into the oviduct because you don't normally see endometrial tissue in the oviduct. Okay, so once they clean the endometrial tissue out of the oviduct, then usually the pregnancy will be normal. Conditions in which the uterus is not able to receive or maintain a fertilized ovum, those are all reasons for female infertility. Okay. Lifestyle changes or hormone therapy can sometimes resolve some of the cases. Others may require surgery, like repairing an obstructed uterine tube. Like a lot of times they'll lavage and clean out what they call curettage, and basically debride all that endometrial tissue out of the oviduct. Others involve direct manipulation or a reproductive process referred to as assisted reproductive technology, ART, and that's been perfected in the past century, in the last half of the century. Okay. Where I went to school, they specialized in that. It always comes to our classes and has students for donors and that kind of stuff. So anyway, assisted reproductive technology. This is basically when you have intrauterine insemination or IUI or artificial insemination. The sperm has been washed and concentrated and then placed directly into the woman's uterus at the time of ovulation. Sperm then must swim up to the uterine tube so that it can naturally produce a pregnancy. It's a very simple procedure done by gamete intrafallopian transfer, GIFT. This is when the sperm and the oocytes are both placed into a woman's uterine tube. Okay. You just do it with both of them, okay? The eggs and the sperm. Fertilization and all developmental steps then must occur naturally in both procedures. Then you can have more sophisticated techniques that allow for fertilization outside the female's reproductive tract. These are called zygote intrafallopian transfer, or ZIFT. The new zygote is immediately placed into the woman's uterine tube. The zygote will then need to develop into a blastocyst and then implant into the endometrial wall. In vitro fertilization. Most or all steps of the pre-embryonic period are completed outside the female's own reproductive tract. So that's why it's called in vitro. In vitro means in the test tube, okay, inside the test tube. In the test tube. Okay, that's what in vitro means. Oocytes are incubated with sperm cells to allow fertilization and the first stages of development to occur. Embryos are then transferred into the woman's uterus, and these then implant naturally. In some cases, to make the embryo, the oocytes are directly injected with a sperm cell or a sperm cell nucleus. Okay? So that's what we refer to as IVF, in vitro fertilization. In many of these assisted reproductive technology procedures, it is possible to use sperm or oocytes donated by third parties to enable the infertile individual or couple to conceive, where the donor may be either known or anonymous to the intended recipient. Recent statistics from the Society for Assisted Reproductive Technology in 2012 indicate the birth rate per embryo transfer to be about 47%. if women are under 35 years of age, and 38% if women are between ages 35 and 37. So the younger you are, the more chances of having positive results with ART. Implantation will occur approximately 4 to 7 days after fertilization. That's when the blastocyst begins to attach to the endometrium of the uterus. So almost a week. Trophoblast then invades the stratum functionalis, the layer of the endometrium, by secreting digestive enzymes that then catalyze reactions to degrade the endometrial lining. Then the trophoblast cells will then divide into 2 layers: a cytotrophoblast, which is the cells that remain intact, and then the outer syncytotrophoblast. The syncytotrophoblast will eventually become a placenta. So the cells, the plasma membranes disappear, and your cytoplasmic contents then merge to form this functionally single large multinucleated cell and the syncytotrophoblast, and it then begins to secrete enzymes to digest the uterine cells almost immediately after formation. So the syncytotrophoblast is responsible mainly for secreting a hormone called ACG, human chorionic gonadotropin. This is sometimes called the hormone of pregnancy because this is what the pregnancy test is based on. It stimulates the corpus luteum and the ovary to secrete estrogen and progesterone, and also helps promote placental development. Corpus luteum will stay around, for 3 more months, though, until the placenta fully develops. Progesterone will suppress menstruation, and it does that by maintaining the endometrium of the uterus and prolonging the secretory phase of the uterine cycle. Remember, once you have that secretory phase, you're producing enough progesterone, which will effectively inhibit LH and FSH being from release, so you're not going to get ovulation. ACG is only produced during pregnancy, okay? So that makes it the basis for the urine pregnancy test. ACG levels will increase in the mother's blood until the end of the 2nd month of development. When that occurs, the membrane called the chorion will take over the role of the corpus luteum in secreting the estrogen and progesterone as a hormone producer. At that point, ACG is no longer needed, and its level will start to decline and remain low through the rest of the pregnancy. At the same time, the inner cell mass, which is going to be the developing embryo, the offspring, will then separate from the trophoblast and then differentiate into 2 layers, the outer epiblast and an inferior hypoblast. And these are what we refer to as the bilaminar embryonic disc. These will become the germ layers, okay? There's also a small cavity that appears within the epiblast. It enlarges to become the amniotic cavity, and that will immediately surround the embryo and fill it with fluid. The bilaminar embryonic disc will also eventually become the 3 germ layers, the primary germ layers. These 3 germ layers are the ectoderm, mesoderm, and endoderm, and these will produce all the tissues in the human body, okay, for future organ development. Remember, ecto means outer, meso means middle, and endo just means inner. Derm, D-E-R-M, means germ, okay, means germ layer, okay? So ectoderm will be the outer germ layer, mesoderm, everything in between. And then the endoderm is the inner lining of the most of the GI tract, the lining of the GU system, the genitourinary system, the respiratory tract. But they'll basically be the inner lining, epithelial linings of most of your body cavities, okay? Whereas the mesoderm will basically produce most of the connective tissues, the bones, blood, and so forth. And the ectoderm will basically produce the nervous system, okay, as well as the outer integument, okay, the skin, okay. So if for some reason the conceptus is not viable, for example, if there's a chromosomal abnormality, it may die. That process is called spontaneous abortion or miscarriage, and the pregnancy will end. But sometimes a blastocyst will implant in a site that's different from the uterus, like in the oviduct, for example. And the pregnancy then continues. That's what we call an ectopic pregnancy. This can be very dangerous, though, for the mother because those can bleed and hemorrhage because any place other than the uterus is not well suited for supporting a developing embryo. Okay. So anyway, there's your implantation stage in the steps that we just described. Okay. Here's your inner cell mass. Here's your blastocyst attaching to the stratum functionalis and the trophoblast differentiating into the syncytotrophoblast and the outer cytotrophoblast. Okay, in here. Here's the inner cell mass in purple. Okay. Okay, that's days 4 through 7. Okay, then day 8, now you've got a epiblast and the hypoblast. The syncytotrophoblast will then digest the uterine wall as the inner cell mass differentiates into the hypoblast and epiblast. Okay, and then on day 12, the syncytotrophoblast now reaches the maternal blood vessels and starts degrading them and degrades them. Okay, and that will basically eventually create little cavities called lacunae. Okay, these little lacunae here. Okay, these little cavities, and the amniotic cavity will then form when within the epiblast. Okay, here's the epiblast here. That's where the amniotic cavity will form. So then on day 10, now the blastocyst is fully implanted. Okay, when it is completely covered now by uterine tissue, as you can see here. It's now completely covered by uterine tissue. And you can see the future 3 germ layers here. Okay, you can see the endoderm in blue, the mesoderm in the red color, and in the endoderm, ectoderm, endoderm, and mesoderm. Ectoderm will become the skin and the nervous system. See if I get this a little bit larger. Okay, so the blastocyst is now implanted, and you have the 3 germ layers. Okay, now you can see a little bit better. Here's your endoderm. Your mesoderm's in the middle. It's going to be all the connective tissues of the human body. And then the endoderm will basically line most of your body cavities. Okay, this would be the endoderm. Okay. So ectoderm, mesoderm, and then endoderm. And this would be your amniotic cavity in here. Placenta out here. These would be, these are going to be where you find eventually the chorionic villi. Okay. So the bilaminar embryonic disc and the trophoblast together produce the extraembryonic membranes. They first appear during the week 2 of development. They continue to develop during embryonic and fetal periods. They include the yolk sac, the amnion, the allantois, and the chorion. Collectively, the membranes protect the embryo and assist with all vital functions, including nutrition, gas exchange, and storage and removal of waste. Of course, the yolk sac will eventually become the source of many of our blood cells. The amnion, of course, is going to produce the amniotic fluid and protects the fetus from drying out, and also provides an environment, cushioning environment. The allantois, that will be eventually the umbilicus, or the blood vessels between the placenta and the mother and the fetus. And then the chorion is the extraembryonic membrane. Okay, and then within the chorion, you'll have the chorionic villi, and that's where your nutrient and gas exchange occurs, in the chorion. So the yolk sac, this arises from the hypoblast cells. Okay? And this is the first extraembryonic membrane to develop. In some animals, like birds, the yolk sac is the main nutritional source for the developing offspring, like chickens. Okay, in humans, the cells from the yolk sac form part of the digestive tract, and they're the source of the first blood cells and blood vessels. Okay, and the yolk sac will also produce the first germ cells, the precursors of the gametes, the eggs and the sperm, which will eventually migrate into the developing ovaries and the testes. The amnion is a transparent membrane that develops from the epiblast. It eventually encloses the embryo in the fluid-filled amniotic cavity. It's only penetrated by the umbilical cord, which contains the vessels that will connect the growing embryo or fetus to the mother's blood supply. It secretes amniotic fluid into the cavity initially, and this fluid is formed by filtering the mother's blood. The embryo's own urine will contribute to the amniotic fluid volume once the kidneys have become functional toward the end of the 8th or 9th week of development. Amniotic fluid has several functions. It protects the embryo from trauma and drying out. It helps maintain a constant temperature. It also allows freedom of movement for the developing fetus, very important for muscle development. And it also prevents the body parts from adhering to each other. The allantois, this is a small outpocketing of the caudal wall of the yolk sac. It develops during the 3rd week of development. In the allantois, this is where you find the early blood vessel formation, and these then become the umbilical vessels, the 2 umbilical arteries and the 1 umbilical vein that will connect the conceptus to the placenta, the lifeline to the mother. Tissue arrest becomes the connecting stalk, which forms the remainder of the umbilical. Inside the conceptus, part of the allantois is called the allantoic duct. It runs in from the umbilicus to the urinary bladder, which it drains. And then the chorion is your outermost extraembryonic membrane. Not the first one to develop, but the last one. It develops from the cytotrophoblasts and the syncytiotrophoblasts. Remember I said those become the chorion. It encloses all the other membranes as well as the embryo. It forms outgrowths called chorionic villi. These will be like the respiratory units, like for the fetus. Okay, these are initially found around the entire surface of the chorion. By the late week 4 of development, first month, the chorionic villi are then blended with the stratum functionalis layer, and they begin to form the principal embryonic part of the placenta. So the chorionic villi will eventually be where the gas and nutrient exchange occurs between the maternal blood supply and the fetal blood supply. So here's the allantois. Okay, here's the allantois as you can see here. This is your future umbilical cord. Okay, the chorionic villi are out here. Villus or villi refers to finger. Okay, villi refers to finger-like. If you hear that term villi, that means finger. So the membrane is the chorion. So that's why they're called the chorionic villi. Okay, anyway, the chorionic villi, that's where you have your oxygen and nutrient exchange with the mother. The amnion, of course, is the membrane, and the amniotic cavity contains the fluid within the boundaries of the membrane. And then you have the embryo proper. This area in the brackets here is the embryo proper, and then this yellow structure is the yolk sac. Remember, the yolk sac becomes the origin of the gametes as well as the early blood cells and blood vessels to form. And then, of course, the outer extraembryonic membrane is the chorion, which out of it comes the chorionic villi. So you can see at this stage in week 3, the chorionic villi are found all the way around the entire surface of the chorion. Okay, and it's not going to stay like that, but at that point, it's what it looks like. So here's the chorionic villi growing more into the direction of the stratum functionalis. Okay, again, here's the amnion, the amniotic cavity, the yolk sac, the allantois, which becomes the umbilical vessels, and the developing embryo. Okay. Now you've got the formation of extraembryonic membranes, and this occurs where the chorionic villi will blend now with the stratum functionalis, as you can see over here. Here's the maternal blood supply over here. Okay, and of course, this is the fetus developing over here, the early embryo. Remember, this is weeks 3 through 8. Okay, so it begins the formation of the placenta. Again, here's your chorion out here, the amniotic cavity, and the amnion. And this is your developing embryo. Okay. Okay, so that's it's late in the 4th week of development, almost a month of development. So anyway, those are your development of your extra embryonic membranes. Okay. Ectopic pregnancy, this is when a conceptus implants and grows in any location other than the endometrium of the uterus. 1 to 2% of all pregnancies are ectopic. Almost all of these are tubal pregnancies, which means they occur in the oviducts. But they can occur in other locations like the abdominal cavity, oral ovaries, or the cervix. Presents a very large risk to the mother because it's the only uterus able to expand. Remember, only the uterus has the ability to expand and sustain a growing conceptus. So when it occurs here, it probably will hemorrhage and could even result in death of the mother unless surgery occurs. And if ectopy does occur, when it does not terminate on its own in a spontaneous abortion, which the majority will, then medical intervention is necessary to remove the conceptus. Otherwise, the region in which it implants can eventually rupture, and it can eventually hemorrhage and possibly cause death of the mother. So it's a very dangerous thing to happen. Okay. Okay, so now we're brought to the embryonic period, week 3 through 8. Okay, the embryonic period, week 3 through 8. So 3 to 8, that's your embryonic period. It begins at the 3rd week of development and continues through the 8th. The blastocyst is fully implanted. The inner cell mass is differentiated to the bilaminar embryonic disc, which has all 3 germ layers, and the extraembryonic membranes have developed. This 5-week stage, the conceptus, we now refer to this to as an embryo. It's not called a conceptus anymore. Our embryonic period starts first with a process called gastrulation, okay, where there's a rearrangement and migration of cells of the bilaminar embryonic disc, and then it forms the trilaminar or the 3-layer embryonic disc. So those 3 germ layers will then become all the major organ systems and process of organogenesis. And that occurs by the end of week 8. Again, since we're going over this, that is a test question. Okay. Placenta also forms during that period, and it begins to provide nutrition and oxygen to the embryo and helps remove any waste. In week 3, the bilaminar embryonic disc now has 2 cell layers, epiblast and the hypoblast. The primitive streak is a little groove on the dorsal side of the epiblast. It elongates along the head-tail line of the embryo, cephalic caudal end, and that's going to establish the head and tail regions, plus bilateral symmetry, right and left sides, and your dorsal and ventral surfaces, your posterior and anterior. So the gastrulation first starts after the primitive streak has completed its development. And then it will start a detachment of the cells from the epiblast layer, and it moves into the primitive streak. And that movement, that migration is called ingression. The first cells that migrate in this way will become the inner germ layer, the endoderm, which then replaces the hypoblast. Next, the cells migrate to position between the epiblast and the endoderm, and this becomes the middle layer, the mesoderm. Of course, the mesoderm will become the connective tissues of the body. Remember, the connective tissues of the body are the most diverse of all the tissues, and they're the most numerous. This includes, your bones, your muscle, your connective tissues, your collagen, blood. Those are all considered connective tissues. And the remaining cells of the epiblast from the outer layer or the ectoderm. Week 3, all 3 primary germ layers have formed. The embryo has now become what we'll call a trilaminar embryonic disc. Ectoderm and endoderm are now tightly packed, and they surround a loosely organized mesoderm. Okay. So that's basically gastrulation and formation of your germ layers. That's the principal function of this gastrulation period. Okay, as you can see here from this diagram. So here's your caudal end, here's your cephalic end. These are the cut edge of the amnion, and this is basically a cross-sectional view. So the epiblast cells migrate, like you see in the arrows, and they ingress, and they migrate into and then under the primitive streak. This is the primitive streak right here. Okay, and those cells then replace the hypoblast and become the endoderm. Okay, and this is the hypoblast down here, and this is the developing endoderm. Okay, so the endoderm will become the lining of the GI tract, the lining of the GU tract, and the lining of the respiratory tract. And then here on the right is a cross-sectional view, okay, of the embryo showing the formation of the mesoderm late in the 3rd week. So there's your trilaminar embryonic disc showing the ectoderm out here, mesoderm, and the endoderm. Okay, so the epiblast cells migrate between the endoderm and the epiblast to become the mesoderm, and the remaining cells then become the ectoderm. Okay. The ectoderm, of course, eventually is going to become the nervous system and the skin, mesoderm and connective tissues, and the endoderm will be the lining of your GI tract and your GU system, as well as your respiratory system. So the flat trilaminar embryonic disc then changes shape during the 4th week of development. You get 2 types of folding, which cause the flat embryonic disc to become cylindrical. That's called cephalocaudal folding, which occurs in the head and tail region. It creates the future head and buttock regions of the embryo. Lateral folding occurs, or transverse, when the left and right sides of the embryo curve and then fold toward the midline. As those sides move close together, the yolk sac is pinched off, leaving only the small endoderm, vitelline duct, left over. The transverse folding then creates a future trunk region and almost immediately forms a primitive gut, which later becomes a digestive tract inside the abdominal cavity. That's really what we're called true coelomates. We have a true body cavity, but it's really basically a cavity within a cavity. Okay, a cavity within another cavity. Okay, the digestive cavity is within the cavity that is your skeleton. Okay, a cavity within a cavity. So that's how gastrulation and the formation of the 3 germ layers occur. Okay. So organogenesis is where 3 primary germ layers differentiate into all the organs and organ systems of the human body. So when that begins, the primary germ layers now have gone through cephalocaudal, head, tail, and transverse folding, which is the bilateral symmetry, and then they begin to differentiate. They specialize. The embryo is about 15 millimeters, or 0.6 inches in length, half an inch in length. When the embryonic period ends at the end of week 8, the embryo will be approximately 25 millimeters, 1 inch in length, and will have recognizable organ systems. Even though not all organs will be functional, some will begin to work. Ectoderm then forms the epidermis of the skin and is responsible for forming the majority of the nervous system and the sense organs, like the eyes and ears and smell, taste, hearing. Cells of the ectoderm then thicken and form the neural plate. It folds inward and then deepens. The edges then fuse to form the neural tube, which then pinches off from the ectodermal layer. The neural tube will eventually become the spinal cord and the brain. Okay, and the anterior end of the neural tube will then become the brain, and the remainder will become the spinal cord. Okay, so that's what we refer to as our central nervous system. By the end of the 4th week, the anterior end of the neural tube develops into 3 enlarged areas called the primary brain vesicles, which are the forebrain, midbrain, and the hindbrain. Forebrain, midbrain, and the hindbrain, as we remember from our days of central nervous system. The neural crest cells now are between the layers of the ectoderm that form the epidermis and the neural tube, and they migrate throughout the embryo to give rise to the cranial, spinal, sympathetic ganglia, and their associated nerves. The pigmented cells of the skin, cells of the adrenal medulla, and the connective tissue cells. So these are all going to be somewhat related, okay, via organogenesis. The mesodermal cells underneath the primitive streak form the notochord, which is your vertebral column. And they serve to support and organize the embryo around a central axis. That produces your central axis, which is your vertebral column. The remnants of the notochord remain in the intervertebral disc as a notochord is then surrounded by the vertebral bones during development. The mesoderm on either side of the notochord then form the somites, these block-like structures. They'll have three regions. The sclerotome will develop into the vertebral bones and the ribs. Okay, the dermatome will develop into the dermis layer of the skin. The dermis layer, remember, is the layer just underneath, just deep to the epidermis. And then the myotome layer will develop into most of your skeletal muscles. So sclerotome, dermatome, myotome. So remember, myo means muscle, derma means skin, sclero basically refers to the vertebral bones. Okay, so those are called the somites. Okay, that comes from the notochord. On the lateral side, the somites, there are clusters of mesoderm called the intermediate mesoderm, and that's where you find the lateral plate mesoderm. The mediate mesoderm forms the gonads and the kidneys, ovaries, testes, and the kidneys. Okay, then the lateral plate mesoderm develops into the spleen, the adrenal cortex, most of the cardiovascular system, including the heart, the serous membranes of the body cavities, and the connective tissue components of the limbs. Okay, so those all come from the lateral plate. Both of them mesoderm. So the endoderm forms the cavity of the middle ear and the auditory tube, the fringe tympanic tube. It becomes the internal epithelial layers of your digestive tract, your respiratory tract, the urinary tract, and your reproductive systems. All of the ventral body cavities that have a lining, okay, which are your digestive tract, your respiratory tract, your GU tract, all together. They form several glands and accessory digestive organs, including the thyroid gland, the parathyroid glands, the thymus for your immune system, your palatine tonsils, and majority of liver, gallbladder, and your pancreas. Okay, so a lot of the organs come from endodermal origin. So by the end of the embryonic period, organogenesis has occurred. In 4 weeks, the embryo now has 3 primary brain vesicles and the somites. By the end of the 8th week, the embryo looks essentially human. Okay, this is at 2 months of age. Okay, all of it with different body proportions. During that period, week 3 through 8, the embryo is essentially sensitive to teratogens. A teratogen is any harmful drug, medication, or agent that's either given by man or a doctor or any kind of healthcare professional that causes damage to the fetus. Those are called teratogens. Okay, substances that can cause birth defects or even death. We used to give, for example, tetracycline and erythromycin to pregnant mothers until we found out it rotted out their teeth. Okay, so a lot of this was done by trial and error and was done by just learning from developmental defects. Unfortunately, that's how it happens. So anyway, all the body's structures will derive from your 3 primary germ layers. Okay. So this is on the left a mini MRI of an embryo at week 4 from 3D imagery that you can see here. Okay, and then on the right you have the embryo at the end of the 8th week. So you start at 3 weeks and you end at 3 weeks. Okay, you can see the amniotic cavity with the amniotic fluid inside. You can see the arms. You can see the fingers. Okay, the eyes. Okay, you can see the buds, the limb buds. Okay, in the weak embryo at 3 weeks, you can see the forebrain, midbrain, and hindbrain. Ear. So ectoderm, endoderm, and mesoderm. Okay, now this is the last period, fetal period. Okay, fetal period is when it really looks human now. Fetal period extends from the beginning of week 9 until birth, and it usually occurs near week 38. Yes, usually when birth occurs. Okay. During pre-embryonic period, the developing embryo then receives the nutrients from uterine milk secreted from the endometrial glands and your digestive endometrial cells from the uterine wall. The embryo also continues to receive nutrition from digested endometrial cells until the placenta is able to function on its own, which is about the 12th week after 3 months. That's the reason the corpus luteum has to stay around for about 3 months in order to provide hormones before before the placenta is fully developed. Okay. But placentation is the process of forming your disc-shaped placenta, which then attaches to the uterine wall and to the embryo or the fetus through the umbilical cord. Placenta is just a temporary organ, but it's a vital organ. And then it's shed after the infant is born, okay? Or it's also the site of exchange of oxygen, nutrients, and waste between the mother and the fetus. Placenta also produces the hormones to support the pregnancy. Remember the hormones, estrogen and progesterone? HCG is secreted early, but once the placenta forms, HCG is no longer needed. Okay. Placentation will begin during implantation, but the majority of placental growth will occur in the fetal period. So here's a fetus at 12 weeks surrounded by the developing placenta, as you can see here. Okay, this is the developing placenta here. Okay, here's the allantois, which becomes the umbilical vessels, and the yolk sac right here. And this is the early embryo. Okay, this is a 12-week-old embryo. Chorion is out here. So the umbilical cord connects the center of the placenta to the fetal umbilicus or belly button. The cord normally contains the 2 umbilical arteries, which, as we know, carries deoxygenated blood away from the fetal heart to the placenta. Now, these will be on the final exam, okay? And you'll have one umbilical vein which carries oxygen and nutrients toward the fetal heart. Remember, they go in opposite directions. Okay, so these are definitely going to be on the exam, okay? Because they're part of development, okay, on the final exam. And I've got a couple of questions that even pertain to these in your medical Jeopardy. The umbilical cord also contains connective tissue called Wharton's jelly, which insulates and protects the umbilical arteries and the umbilical vein. And it's also covered externally by the amniotic membranes. So when the embryonic period begins, the blastocyst is now implanted in the uterus, and the chorionic villi have penetrated the uterine blood vessels to form the lacunae, the cavities filled with maternal blood. And they merge into a single blood-filled cavity called the placental sinus. But once placentation has begun, now the stratum functionalis of the uterus is called the decidua. Decidua, the term decidua, okay, means temporary, okay? Decidua means temporary. Literally, in Latin, means temporary. You know, like when you look at deciduous trees, they're just temporary trees, like pine trees are deciduous trees, whereas live oaks are permanent, and they're going to stay around a while, okay? Anyway, the region of the endometrium that lies beneath the fetus, that's called the decidua basalis. And the region that surrounds the uterine cavity forms the decidua capsularis, okay? So this will be the umbilicus with the umbilical veins, the one umbilical vein which carries oxygenated blood, that's the color red, and the 2 umbilical arteries which carry blood from the fetus. So the deoxygenated blood that's going through the 2 umbilical arteries is going this direction. And then the blood from the umbilical artery, the blood from the umbilical vein, of course, is going in this direction, okay? That would be your umbilical artery, your umbilical vein. Your umbilical vein is going to carry oxygen to the fetus. From mother to the fetus. From the maternal artery to fetus via chorionic villi. Via the chorionic villi. So it's a unique organ because it develops from both fetal and maternal structures. The fetal origin would be the chorionic villi, and the maternal origin would be the decidua basalis. Remember, the decidua basalis was at one time the stratum functionalis. At one time, we called that the stratum functionalis, which is what's shed in menstruation. So the chorionic villi are then filled with blood vessels carrying fetal blood and surrounded by maternal blood in the placental sinus. Remember, the blood supplies do not mix. The placental barrier is composed of a basement membrane and a thin layer of cinctular trophoblast, and that keeps the blood supply of the mother and the fetus separated, so you don't get autoimmune problems, or immune complexes forming. So since your maternal and fetal blood supplies are so close, some substances are exchanged across the placental barrier between the fetal blood and the maternal blood supplies. Your oxygen and nutrients will diffuse from the maternal blood into the fetus for transport to the tissues, whereas waste products from the fetus, mainly your nitrogen waste products like ammonia and urea, those will come from the fetus to the maternal blood for removal. So the mother's going to have double duty. She has to get rid of not only her waste, but the fetal waste. So here's the schematic drawing showing the movement of substance across the placental barrier. As you can see, oxygen and nutrients are freely diffusible from the maternal blood in the placental sinus across the basal lamina, okay, and into the fetal blood in the capillary. Here's the cytotrophoblast here, and here's the cinctiotrophoblasts here, okay? So these make up part of that placental barrier. Here's the fetal endothelial cell and the fetal basal lamina. And here's the maternal basal lamina and her blood capillary over here. So oxygenated maternal blood flow to the decidua basalis layer of the uterus and then through the maternal arteries into the placental sinus. Maternal blood then touches the placental barrier that allows diffusion of substances, but does not allow passage of blood cells. It's kind of like your blood filtered in your kidneys across the glomerular filtration membrane, very similar to that. But once the diffusion occurs, the maternal blood, which is deoxygenated and carrying waste, will flow into the maternal veins and back to the mother's cardiovascular system. Okay, so the mother has to really eliminate the waste products via the placenta. So the deoxygenated fetal blood then leaves the fetus via the two umbilical arteries and then enters the two and then flows into the placenta and moves into the fetal capillaries and the chorionic villi, but that's on the fetal side of the placental barrier. Fetal blood then picks up oxygen and nutrients from the mother's blood and delivers waste to the mother by diffusion. But again, no blood cells move through the barrier. Also, you must remember that fetal hemoglobin has a much greater affinity for oxygen than adult hemoglobin. Okay, and that allows us to pull oxygen away from the mother's hemoglobin at that placental barrier, and that's the reason the fetus has fetal hemoglobin. The oxygenated blood then leaves the placenta, then flows back to the fetus through the single umbilical vein. So that hemoglobin will just grab onto that oxygen very rapidly, because it has, I think it's like 1,000 times more attracted to the oxygen than what adult hemoglobin is. It's amazing how everything has a purpose. That's why life is such a miracle. In addition to nutritive functions, the placenta also functions as an endocrine organ. It stimulates production of the hormones like HCG, and also as the placenta grows and results in then a subsequent reduction of the cinctiotrophoblasts. Corpus luteum will also relinquish production of progesterone and estrogen to the placenta by the end of the 3rd month of gestation. What else does the placenta produce? What other hormone? Okay, and another substance that's produced by the placenta is a substance called human placental lactogen, HPL, and also placental prolactin. Remember, we have prolactin from the anterior pituitary, but we also have prolactin from the placental source. So that helps prepare the mammary glands for production of milk, HPL. Relaxin is another hormone that relaxes the body's muscles, joints, and ligaments. And mainly this is to facilitate stretching during the delivery of the newborn. This is all meant for labor and delivery, a relaxing hormone. Unfortunately, placenta previa is like, it's an unfortunate thing to happen. What happens is that either part or all of the placenta that attaches to the inferior portion of the uterus, very near or even covering the cervix. Most cases result in live births, but it may cause a spontaneous abortion or a premature birth. Also very dangerous to the mother because of hemorrhaging, because the uterus stretches to accommodate both the fetal and the placental growth. And therefore the placenta may hemorrhage or become infected. And that increases the risk of maternal mortality. This is one of the common reasons for mothers that are, especially were born in the 20th century to die during childbirth. Typically, C-section, surgical removal of the fetus, was preferred method of delivery in cases of placenta previa. So basically, in fetal development is your maturation of your tissues and organs. Fetal growth is very rapid, though, and the fetal size will increase dramatically. Okay, common fetal measurements of length are what we call crown rump length, CRL. Embryo, for example, is about 25 millimeters, 1 inch length, and it can grow to be as large as 50 centimeters, which is about almost 20 inches in length during the fetal period. So you're going from 1 inch to 20 inches. Okay. Weight will also increase, most obviously in the last 2 months of development. The average weight of full-term fetus usually ranges from 3.2 to 3.5 kilograms, 2.2 pounds per kilogram, so that'd be about 7 to 8 pounds. 2.2 pounds per kilogram, so 2.2 times 3.2 would be about 7, and 2.2 times 3.5 would be about 8. Month 3, the body lengthens as the head grows. As the head growth slows, the upper limbs also grow to their birth length. And this is also when you see ossification beginning in your bones, month 3 of gestation, which is the eyes are still not. The eyes are well developed, but on the lateral side of the head, the eyelids are still fused. The bridge of the nose will develop, and the external ears are present but not prominent. The genitals are now distinguishable at 3 months as either male or female. So by the end of the 3rd month, the approximate crown rump length is about 9 centimeters, 3.5 inches. So month 3 is when ossification, genitals, so you can tell the sex of the baby, of the fetus. Month 4, the body growth is still rapid. The lower limbs will lengthen, and the joints are forming now as your skeletal tissues continue to mineralize, ossify. The fetus now will have nipples and some hair, okay? Even though it's the nudo hair, okay? By that point, the kidneys are well formed. At month 4, digestive glands are still forming, and the heartbeat can be heard with the stethoscope at month 4. So about 4 months, just past the trimester, first trimester, you can actually take a stethoscope and listen to the heartbeat. So it also begins to develop reflexes. Okay, that's the reason when you, and also will startle and turn away from loud noises and bright lights. Okay, approximate crown rump length is about 5.5 inches by the end of the 4th month. It also can, it also experiences. Month 5, that's when growth will slow down, and lower limbs will achieve their correct proportions relative to the body. Okay. The hair will also start to grow on the head. The skin now is covered by what we call the nugo, or downy hair, and also a secretion called vernix caseosa. Okay, and it's basically a white to grayish color secretion, mainly composed of shed epithelial cells and sebum from the sebaceous glands of the skin. That's called vernix caseosa. Both lanugo and vernix caseosa will cover the skin, protect it from the amniotic fluid. Month 5, now you have brown fat being produced, which helps with heat production after the fetus is born. Mother can also feel the movements, the quickening, as the skeletal muscles begin to contract. That's called quickening. Approximate CRL is about 7.5 inches by the end of the 5th month. They use a crown rump length because that's the easiest to measure with the sonogram. So here's development during the fetal period, where this is actually the actual size of the fetus. So this is a fiber optic view of the fetus at month 3 on the left. This is a fiber optic view of the fetus at month 5 on the right. See the fingernails? Okay, that's at month 5. Fingers, lips, nostrils. You can even see what it's going to potentially look like. Okay. Okay. Month 6, the fetus gains significant weight. Eyebrows and eyelashes also start to appear at month 6. Eyelids, which have been fused since 3rd month, are now partially open. Skin is wrinkled and translucent. Lungs begin to produce surfactant, and that's important for survival if the infant happens to be born prematurely, which sometimes happens at 6 months. Approximate CRL is about 9.1 inches, 23 centimeters by the end of the 6th month. CRL means crown rump length. Distance between the crown and the rump, and the distance between the rump and the leg. Okay. So the distance between the crown, the cephalad, and the tail. Okay. Eyelids now start opening completely at month 7. Fat also is deposited in the subcutaneous tissue, so the skin is going to become a little bit smoother than at month 6, but still wrinkled and red. Fetus usually turns upside down at month 7, assuming the vertex position, which you hope it stays that way. In males, the testes will then begin to descend through the inguinal canal and into the scrotum. By the end of the 7th month, approximate CRL is about 28 centimeters, about 11 inches in length. And then 8th month and 9th month, that's when the fetal neurons form networks. These nerve plexuses, blood cells form in the bone marrow. Digestive and respiratory systems also complete development during the 9th month. Fetal skin is less wrinkled and the nugo is shed. In males, the testes will complete their descent into the scrotum. Approximate CRL is about 30 centimeters, 12 inches a foot by the end of the 8th month, and about 36 inches, 14 inches by the end of the 9th month. So about 266 days, the average day number of days for gestation, the fetus is considered full term, okay, after fertilization and ready to be born. Scalp usually has hair, fingers and toes are well developed. Nails, if however the fetus is born before that time, its tissues and organs may not have developed completely, okay? So sometimes, especially the liver, they may have to be given treatments, radiation, sun lamps and things like that, ultraviolet lamps, sun lamps, things like that, to provide a little bit more time for the organs to mature. So the fetal circulation in the cardiovascular system will change rapidly soon after birth, and mainly to adjust to life without a placenta. And being in the most conspicuous and anatomical changes between the placenta and the postnatal state, mainly occur in the cardiovascular system. Because remember, you're basically going from a heart that have functioning lungs and where all the oxygenated blood comes from the mother to an independent heart that basically has two separate blood supplies, a pulmonary circuit and a systemic circuit. And that's not really true for the fetal circulation because most of this oxygenated blood comes from the placenta. So you have to have a few structures that are going to have to accommodate birth. So unique cardiovascular structures present during prenatal development include the two umbilical arteries and the umbilical vein, and three circulatory shortcuts called the vascular shunts. We've already gone through these a little bit in the first half when we looked at the cardiovascular system. Again, this should be just review for you, hopefully. So the ductus arteriosus, the ductus venosus, foramina valley, and the ductus arteriosus. Remember, the ductus venosus allows blood from the umbilical vein to bypass the liver, and then connects to the inferior vena cava, and then flows into the right atrium of the heart. The ductus venosus will eventually become a ligament off the liver. Okay. The foramina valley, which is the foramen in between the right and left atrium, which allows the right atrial blood to communicate with the left atrial blood, will close also. It becomes a depression called the fossa ovalis after it closes. Okay, so the hole in the interatrial septum that directly connects the right and left atria allows blood to skip ahead and avoid the pulmonary circulation by moving directly from the right heart to the left heart and then into the systemic circulation. So that's going to close and become the fossa ovalis. And then the ductus arteriosus, that short passage that connects the pulmonary trunk to the aorta, which allows blood to move from the pulmonary trunk directly into the aorta. Because remember, the pulmonary trunk is not really taking blood to the lungs to be oxygenated. Okay, that's not the function yet. Okay, so it just goes straight into the aorta. So after birth, the shunts and the umbilical vessels close, and then the normal circulatory pattern begins within a year in most infants. Your flaps in the interatrial septum will seal, and the foramina valley becomes a depression called the fossa ovalis. Resistance and pressure changes in the pulmonary trunk and aorta will cause the ductus arteriosus to close, and that will eventually occur completely approximately 3 months after birth. The lid of the ductus arteriosus is the permanent ligamentum arteriosum. Okay, the permanent cord called the ligamentum arteriosus. If you remember what it looked like, remember the pulmonary trunk went up like this, and then you had the aorta behind it, which went like this. So you have this little duct between the pulmonary trunk and the aorta. Okay, and that connected the pulmonary trunk and the aorta. Here's the pulmonary trunk. Put that in blue. There's your pulmonary trunk because it's carrying deoxygenated blood, so I'll color that blue. And here's the aorta down here. This is going to carry, which is going to carry blood to the systemic circuit. So I can draw this a little bit better. And here's the aorta. And then you got your pulmonary trunk over here. And then, of course, remember, it separates the pulmonary arteries, which go to the lung shield. So you really don't need the ductus arteriosus, okay? So this is the ductus arteriosus. ductus arteriosus, and that's going to close it at birth. And it takes about 3 months for it to completely close. Okay, that basically closes at birth and becomes the ligamentum arteriosus. Abbreviate there. Anyway, so the ductus venosus will also degenerate and become the fibrous ligamentum venosum, and the umbilical arteries become the medial umbilical ligaments, and the umbilical vein becomes the ligamentum teres, or the round ligament of the liver. Okay, so the umbilical vein becomes the ligamentum teres, the umbilical arteries become the medial umbilical ligaments, and the ductus venosus, which of course bypasses the liver. Remember, you have your liver. Here's your liver. Let's see if I can draw it here. Here's your liver. And you've got your umbilical vein called the ductus venosus, basically bypassing the liver and going straight into the inferior vena cava. So here's your ductus venosus, okay. And basically it goes into the inferior vena cava of the fetus. So here's the inferior vena cava. The liver is over here. Okay. Okay. Anyway, this ductus venosus that you see right here, ductus venosus is going to eventually going to close, and it becomes the ligamentum teres. It becomes the ligamentum teres, or also called the round ligament of the liver. It's located on the ventral side of the liver. Okay. So anyway, these are your fetal cardiovascular structures that are unique. And as you can see, you got your ductus arteriosus that closes and becomes a ligamentum arteriosum. The foramen ovale closes between the right and left atrium. It becomes a fossa ovalis, that depression in the wall of the right atrium. The ductus venosus over here, there's your ductus venosa, which is really a continuation of the umbilical vein. It's going to close, okay, and it becomes the ligamentum teres, this little ligament that you see here, okay, on the ventral side of the liver, okay. And then the umbilical vein over here, it becomes a ligamentum teres, and then the two umbilical arteries become the medial umbilical ligaments, okay. They become these two umbilical ligaments that flank the bladder, okay. So I'm not going to talk about prematurity, or just real briefly, just won't talk very much about it though. Most common respiratory problem for prematurity is IRD, okay, infant respiratory distress, and it's mainly because of inadequate surfactant. Because if they're born before 24 weeks gestation, surfactant is really not produced in the alveoli of the lungs until about 3 to 4 weeks. Without surfactant, the alveoli collapse during insulation, and they're difficult to reinflate due to the alveolar surface tension. So synthetic surfactants can also help premature infants breathe until their lungs can develop sufficiently. That's why they have to stay in the hospital for a number of months sometimes, if they're born prematurely. Other difficulties are they have minimal sucking and swallowing reflexes and require feeding tubes. Underdeveloped liver cannot produce enough clotting factors for albumin, so they bleed easily and may develop swelling, edema. Hypothalamus really is not completely developed. It cannot maintain a constant body temperature, therefore they have to be kept warm in the incubator. Almost 30% of infants are born in 23 weeks of pregnancy. Okay, 50 to 60% of those are born in 24 weeks. Even though neonatal care has increased survival rates, we still have these statistics. About 75% are born in 25 weeks. More than 90% are born in 27 to 28 weeks. Infants who survive often face lifelong issues such as cerebral palsy, learning disabilities, and hearing and vision loss. Okay, so they may survive, they're still going to have permanent condition that they have to face. Okay. So anyway, that's basically all we're going to cover on chapter 27. Okay, so I want to go to some reviews now. I put the lab practical review on video, and I did a pretty good presentation. So I'm going to let y'all just view that on your own. But I would like to go through some of the medical jeopardies. Let's do the urinary system first. Okay, the urinary system. We'll start with the urinary system. Tube that carries urine from the kidney to the urinary bladder is the Anybody want to take a guess on that one? What's the answer to this one? Anybody? Yeah, ureter. Very good. Very good. Got one brave soul there. The renal medulla is also called what's located in the medulla? Renal pyramids. Which of the following blood vessels are associated with the nephron? Okay, we're talking about within the nephron. Well, all of these, arcuate, interlobar, cortical, remember, these are not associated with the nephron. They go up to the nephron. Efferent arteriole is the only one that's associated. Okay, efferent arteriole is the only one that's really associated with it. Okay. Which of the following blood vessels are associated? Okay, efferent arteriole. Which of the following empties urine directly into the renal pelvis? Well, here we're testing your knowledge of your urine draining structures. Remember, your nephrons collect the urine, after they've gone through the different tubules, proximal distal tubule, and they go into the collecting ducts. Finally, the papillary collecting duct, we call it urine. Okay, so the minor calyx, remember, it drains the renal pyramid. Okay, and so if you have 9 renal pyramids, that means you have 9 minor calyxes. Okay, and then the minor calyxes then drain into the major calyx. And then the major calyxes then drain into the renal pelvis. Okay, so the major calyx would drain into the renal pelvis. Okay, make sure you remember, just all that requires is for you to know the anatomy of the collecting structures. So renal, papillary, collecting duct, minor calyx, that leads to a major calyx, that leads to the renal pelvis, renal pelvis, and leads to the ureter. And that then leads to the bladder. And the urinary bladder then leads to the urethra. That's your entire collecting. Renal pathway collecting duct, minor calyx, major calyx, renal pelvis, ureter, bladder, and then urethra. Okay, that's your order there. Okay. Renal ptosis is like ptosis of the eyelid is like when you lose part of your eyelid closing. Well, ptosis of the kidney is when you lose your, yeah, that's right. Fatty encasement around the kidneys, yeah, is lost. That's right. Reduction of the fatty encasement around the liver. Nephrons are found where? Medulla, cortex, capsule, or sinus. Mm-hmm. In the cortex. They would be in the cortex, in the renal cortex. Capsule is just the covering. Capsule is your connective tissue covering. But it'd be the renal cortex. Remember, you have two divisions. Here's your kidney. There's a ureter coming out. So out here, this outer region, and that's the cortex. And then the middle region is the medulla. Where you find the pyramids, where you find these little pyramids. That would be your medulla. And then the pelvis is down here in the middle. So that would be the cortex. Which of the following can be evidence of glomerular inflammation? Well, yeah, protein. That's right. That's exactly right. Protein is right. And the reason for that is when you have glomerular inflammation, that means that's usually from an infection, like a UTI. And it usually is from an inflamed glomerular capillary. Okay, so the filtration membrane is defective, okay, and start losing selectivity and proteins and even cells can show up in the urine. That's because when you have inflammation, that makes the membrane very permeable to cells and proteins. Okay, that's why you see them in the urine. So right, D is right. A decreased amount of ADH leads to an increase or decrease? Yeah, an increase in amount of urine. Because remember when you lose ADH, remember ADH stands for antidiuretic hormone. That means it's going to prevent diuresis, right? So if you don't have ADH, you don't get water reabsorption, okay? So all the water stays in the kidneys, okay? So you're going to be going to the bathroom all the time when you have a decreased amount of ADH. And that's why people that drink a lot of coffee, because caffeine and also when they drink a lot of alcohol, Alcohol and caffeine are ADH inhibitors, okay? And that's the reason you go to the bathroom all the time when you drink a lot of coffee or drink a lot of alcohol, okay? Because they're ADH inhibitors, okay? Inhibit ADH and you prevent those collecting ducts from being permeable to water. So all the water stays in the collecting duct. So that's the reason you go to the bathroom all the time. Remember, you have to have ADH to make the collecting ducts permeable to water, okay? So increased amount of urine would happen. The renal corpuscle consists of two things in a renal corpuscle. Glomerulus, which is the capillaries, and a capsule. Okay, those two things, form the renal corpuscle. Okay, both of these form the renal corpuscle. Both B and C. Which of the following is not a structure of the nephron? Well, we know Bowman's capsule is nephron loop. Yeah, papilla is not a, yeah, right, exactly. Which of the following are used to move sodium out of the nephron? Well, GFR would be, glomerular hydrostatic pressure. Remember, GHP is what drives fluid across the membrane, so that would definitely move sodium out of the nephron. Ion pumps utilizing ATP, well, that's how we get your sodium out of the cell and into the extracellular fluid. So that would be true. So all the above would be the answer to that one, because you probably already knew that one. ADH is a hormone that is active where? Yeah, distal tubule and collecting duct. Distal tubule and the collecting duct. The primary force pushing water and solutes out of the blood across the filtration membrane is the design and size of the podocytes, the thickness of the capillary endothelium, the net filtration pressure. Well, the main force pushing water in solutes is your net filtration pressure. Okay, remember your NFP. Just refresh you on that. NFP is equal to your glomerular hydrostatic pressure, GHP, minus the glomerular colloid osmotic pressure, which pulls water back into the capillary, added to your capsular, what's in the glomerular capsule hydrostatic pressure. Okay, remember, capsular hydrostatic pressure also pulls water back into the capillary. So you take the sum of those two and then subtract it from your glomerular hydrostatic pressure, and that gives you your net filtration pressure. Okay, so if your normal glomerular hydrostatic pressure is about 55, okay, it's usually about 55 millimeters of mercury. So 55 millimeters of mercury minus a normal colloid osmotic pressure is about 30, okay, 30 millimeters of mercury, okay, and then normal capsular hydrostatic pressure. The capsular hydrostatic pressure is what's inside the glomerular capsule where the fluid collects, and that's going to be fairly low because you only produce so much capsular fluid at a very slow rate because it's just forming. So it's going to be fairly low. It's going to be about not really more than 5 millimeters of mercury, 5 to 10 millimeters of mercury. So probably about 5 millimeters of mercury. Okay. So you take, or it ranges between 5 and 10. So, but we'll say 5, because that's probably more likely. So you take 50 minus 30 plus 5, or 50 or 55 minus about 35, or that would give you your net filtration pressure. Okay, but in this case, it would be about 25 millimeters, about 20 millimeters of mercury. But most net filtration pressure is about 10. Okay, net filtration on the average for most healthy kidneys is around about 10 millimeters of mercury. That's an average. But you take the sum of the capsular hydrostatic pressure plus the glomerular colloid osmotic pressure, and then subtract that from the glomerular hydrostatic pressure. Okay. But yeah, it's your net filtration pressure. Autoregulation locally of the kidneys is done by remember the myogenic mechanism? That affects mainly by the afferent arteriole and the macula densa in the distal tubule. Afferent arteriole and the macula densa. Yeah, that'd be B. That's correct. Which of the following is an ADH inhibitor? Like we said a while ago, alcohol is an inhibitor, exactly. Which term below concerning urine formation describes the movement of molecules in the nephron from the blood? When you go from the nephron into the blood, what do we call that? Reabsorption, right. When you go from the filtrate to the blood, we call that reabsorption. Filtrate to the blood, we call that reabsorption. Secretion would be from the blood to the filtrate. That would be the opposite direction. Reabsorption, going from the nephron, you're basically just got the movement of molecules into the nephron. I'm sorry, secretion. I'm sorry, I read that wrong. You're right, secretion. Secretion, yeah. Moving molecules into the nephron from the blood, secretion. Okay, reabsorption is just the opposite. That's my fault. I read that wrong. Movement of molecules into the nephron from the blood, secretion. You're exactly right. Secretion is correct. Okay, water that is absorbed in the distal tubule and the collecting duct is considered what? Well, if it gets to the distal tubule, that means all the obligatory reabsorption has occurred already. Okay, and obligatory means just it occurs by the factors like osmosis. You got an osmotic gradient that's going to pull the water out. Okay, but by the time it gets to the distal tubule, remember hormones are going to determine the rest of the sodium and water movement. So that's called facultative. Okay, so that would be facultative reabsorption. Facultative means it involves hormones. Involves hormones present. Like the hormones that we're looking at are aldosterone and ADH. And I guess I should mention A&P. A&P is a third hormone. But I always forget A&P. A&P works the opposite of aldosterone and ADH. So. Excretion of a concentrated urine requires overhydration, aquaporin channels, thiazide diuretics, or lack of aldosterone. Well, these two really don't have anything to do with the concentrated urine. Overhydration, you would get a dilute urine. With overhydration, that would result in a dilute urine. Okay. And the other two, these really don't have any relation. Thiazide diuretics, these make you lose your potassium. Thiazide diuretics, that make you lose all your potassium. Lack of aldosterone, yeah, you won't get your sodium reabsorption, and therefore your water reabsorption. So it actually would be more dilute, okay? It would be more dilute. So the only one that would work on this would be B. So B would be the answer on that. You have to have open aquaporin channels, okay? So that the ADH will cause the water to come out of the collecting duct and back into the blood, Okay, and that makes the urine very concentrated because all the water left the urine to go into the blood. So, but you need open aquaporin channels. In distal tubule, changes in sodium concentration in the tubular fluid are monitored by what? Macula densa. Those are the macula densa cells. That's right, macula densa. Remember, that's part of that tubulo-glomerular feedback mechanism. Tubulo-glomerular feedback mechanism, where you get your sodium levels dropping. That can signal the afferent arteriole, and that will cause release of renin. Renin will cause the production of angiotensin II, and that'll cause increased sodium and water reabsorption. Okay, but it's usually low sodium. Okay, but they're monitored by the macula densa mainly. In mature reproductive female ovaries, what happens? Secondary follicles develop in response to FSH, numerous ovarian, both meiosis division produce an ovum. Millions of primary oocytes develop from ovum study. Which one of these is the only true statement that they've got here? You don't get numerous oogonia. You're only going to get like a few hundred at the most. Okay. Both meiosis will produce an ovum? No. Okay, that's definitely not true. Okay. Remember, the second meiosis division is really like mitosis because you're just separating the sister chromatids. Okay, so you're only producing an ovum with the second. Yeah, A is right. That's right. The only true statement is the secondary follicles develop in response to FSH. That's correct. Why is glomerular blood pressure slightly elevated? Yeah, A and C is correct. Actually, A and C is correct. D would be correct on that one. See, but the afferent is slightly wider than the efferent arteriole. That is true. Okay, and that fact makes the pressure a little higher. Okay, if you have one vessel a little bit wider than the other, that's going to cause the fluid to back up. Okay, because the other vessel is more narrow. Okay, and that increases pressure. Okay, but it also is because you got two arteries. On either side, you got an artery on one side of the capillary and another artery on the other side of the capillary. Remember, most capillaries will have an artery on one side but a vein on the other. Okay, whereas in the capillary beds, in the kidneys, the glomerular capillaries, you got two arteries. Okay, you got an artery on one side feeding the capillary, and you got another artery on the other side draining the capillary. Okay, and that raises the pressure. That's the reason you have a glomerular hydrostatic pressure of 55. This is why your glomerular hydrostatic pressure is 55 millimeters of mercury. There are most organs, the hydrostatic pressure is about 25 millimeters of mercury. About 25 millimeters of mercury. Okay. So, but both A and C are true. Okay. Both A and C, that would be true. What substance produced from the kidneys when a decrease in blood pressure occurs? From the kidneys. Well, we can rule out some of these. Angiotensinogen. Okay, angiotensinogen is produced by the liver. Angiotensin II, remember, is converted from angiotensin 1 in the lungs. Okay, by angiotensin converting enzyme. So we can rule those out. Those don't come from the kidney. Okay. Yeah, renin is correct. That is correct. Simone, you are correct. Very good. Lube diuretics like Lasix affect which portion of the nephron? That would be your nephron loop. Okay. In fact, if you go into the medical field, you will learn about this a lot. Okay. Because when they get on these loop diuretics like Lasix, they eventually wear out their nephron loop sometimes. You know, you have to be careful when you put somebody on a diuretic, but it affects the loop, the nephron loop. Okay. And that's the reason you have to give them potassium, because a lot of times they'll have a low potassium level because they're on Lasix. Okay. Causes low potassium levels. A lot of times you have to give them supplements of potassium. But they're nephron loop, they're loop diuretics. Okay. The renal cortex, okay, I think we already talked about that one. It contains glomeruli. Remember, it contains glomeruli, it contains Bowman's capsule, and it contains proximal tubules, all three of these. Okay. What controls the sodium reabsorption of the distal tube and collecting duct? Aldosterone. Aldosterone. Remember, aquaporins are in the collecting ducts. Okay. Of course, calcitonin is basically lowers your calcium. Okay, calcitonin basically lowers your calcium. Parathyroid hormone raises your calcium. Doesn't really have anything to do with sodium, though. What type of nephron plays an important role in producing concentrated urine? Remember, most of your nephrons are cortical, but there are certain nephrons that are very important for producing concentrated urine. Which of these is very important in producing concentrated urine? Yeah, medullary, juxtamedullary, you're right. Juxtamedullary, that is correct. Podocytes are part of what? The filtration slits, the juxtamedullary complex, the glomerular capsule, the proximal tubule. Remember, the JGA is the juxtaglomerular complex is the macula densa of the distal tubule and the afferent and efferent arteriole. Okay. Glomerular capsule, of course, as a visceral and parietal layer. Okay. Podocytes produce pedicles that produce the filtration slits. Okay. But they're part of the capsule. Okay. The podocytes are part of the glomerular capsule. They form the visceral layer. They're part of the visceral layer of the capsule. Remember, the visceral layer is the podocyte cells and their pedicles with filtration slits. Which region of the nephron has abundant microvilli? Anybody? Proximal tubule. Very good. Proximal tubule. You're right on. Which of the following conditions would increase the GFR? An increase in colloidal osmotic pressure, an increase in glomerular hydrostatic pressure, an increase in capsular hydrostatic pressure, or all the above. Remember, capillary hydrostatic pressure goes toward the capillary, okay? So that definitely wouldn't increase GFR because that's going in the opposite direction, okay? So that rules out the last two. So which of these two would be true? An increase in colloid pressure or an increase in GHP? Remember, when you have GHP, exactly. That's right. Correct. You're exactly right. Colloid osmotic pressure, remember, that moves in the opposite direction, okay? Remember, when you're looking at your blood capillary, okay? Okay, remember your blood capillary, remember your GHP goes in that direction, okay? Here's your oops. Here's your capsule, okay? And then, it forms, there's your tubule. Okay, so here's your capillary here, okay? So your GHP goes in the forward direction, okay? Your colloid osmotic pressure, COP, it goes in the opposite direction. It goes back toward the capillary, okay? And as well as your capsular hydrostatic pressure, it also forces it in the opposite direction. It also goes toward the capillary. The only one that goes in the forward direction is your GHP. So if you increase your GHP, you're going to increase your glomerular filtration rate. Remember, normal GFR, make sure you know the normal GFR. Normal GFR is 125 mils per minute. Okay, so 60 mils per minute means renal failure. Okay. Because when you get half that rate, you're going to go into renal failure, and then when you do go into renal failure, then you become uremic. Uremia develops. Toxic metabolites. Toxic metabolites like ammonia. Okay. And eventually, you're going to need a kidney transplant or dialysis, or you're going to die. Okay. So, which of the following substances would not be found in normal filtrate? In other words, which one effectively blocks it from going into the nephron? Yeah, albumin. You would not see albumin in a filtrate. You can see glucose, especially in diabetics, potassium, and of course, most of your urine is urea. 40% of the urine is waste products. But albumin is an amino acid. It's a small amino acid, so it forms a protein. Okay, so you wouldn't see, hopefully you don't see albumin. Which is the following would be a result of increases in blood pressure. Okay, remember that's called the myogenic effect. That's what we refer to as the myogenic autoregulatory mechanism. And your blood pressure goes up, your afferent arteriole is basically constrict. Okay, your afferent arterioles constrict when your blood pressure goes up. And what that does, remember, like we were saying in the lecture, think of your nephron, think of the glomerulus as a kitchen sink. Okay. The faucet of the sink is the afferent arterial. The drain plug of your sink is the efferent arterial. So the afferent arterial constricting, afferent arterial constricts is similar to turning the faucet handle so that you reduce water flow into your sink, so that you reduce water flow into your kitchen sink. Okay, and on the other hand, constricting the efferent arterial is similar to putting the stopper in your sink to keep water from draining, keep water from draining out. Okay. And if you think of it like that, you remember that if you have an increase in systemic blood pressure, the afferent arterioles constrict, and that would be a result of increase in systemic blood pressure. What is the NFP if HPGC is 60, OPGC is 35, HPCS is 15? All you do on this situation is you take 60 millimeters of mercury, 60 minus 35, which is the opposite direction, plus 15. Okay. Or 60 minus 50. 60 minus 50, which equals, 10 millimeters of mercury. And that's your answer. That's right, 10 millimeters of mercury. That's how you find out, that's how you determine your net filtration pressure. That's right, 10 millimeters of mercury. Which of the following glomerular filtration mechanism involves the mechanism cells? Glomerular, hormonal, myogenic is a constriction. Okay, myogenic. That's when your diameter changes. Your diameter of vessel changes. Okay, like in vasoconstriction with increased blood pressure. Tubular feedback, that requires your macula densa, monitoring sodium levels. Okay. So tubular feedback would be the macula densa. Okay. Most of the nutrients in the filtrate are absorbed where? Proximal, distal, or nephron loop? Proximal distal tubule. That's the proximal tubule is where most of your volume is going to change. Remember, 65% 65% of water, sodium, potassium, chloride, 100% of glucose, amino acids, are reabsorbed in the proximal tubule. That's where you're going to see the greatest volume change. The greatest volume change is in proximal convoluted tubule. Proximal tubule. Most of the nutrients infiltrate or reabsorbed in the proximal tubule. In the distal tubule, a blanket secretes for every sodium that is reabsorbed. What's secreted for every sodium? Yeah, potassium. Potassium, exactly. Potassium. Exactly right. There's potassium as well as a hydrogen. Potassium and hydrogen, for every sodium that's reabsorbed, there's potassium and a hydrogen that are secreted. When glucose spills over into the urine, it is what? Well, you only got so many carrier proteins. You only got so many transport proteins that can carry glucose. Even though you may have enough insulin, you only have so many transport proteins that can carry. So if you saturate all your carrier proteins, you're not going to be able to carry any more glucose into the cell. So the glucose starts spilling out into the urine. So you've exceeded your transport maximum. Okay, when that happens, you've exceeded your transport maximum. And then glucose starts going into the urine. Glucose spills into the urine. That's why you see it in diabetes. It's what's seen in diabetes mellitus. If your urine contains fewer potassium than your filtrate, then some potassium has been what? Well, if you've got more, if you've got more potassium in your plasma than in your urine, that means something happened with the potassium as it went through the nephron. So it was filtered and reabsorbed. Yeah, exactly. It's filtered and reabsorbed. Exactly. That's exactly what would happen. Hormone-controlled sodium resorption is controlled by aldosterone and occurs in the distal convoluted tube. Okay, that's just something you've got to remember. Okay. Which of the following would be an abnormal constituent of urine? Again, we said albumin you wouldn't see in your urine. The collecting ducts are permeable to water when the hormone ADH is present. Okay. Okay. Which of the following would be an abnormal pH? Your pH is going to vary between pH, remember, it's how much hydrogen. pH will vary between 4.8 and around 8.9. Okay. So 11 would be way too high. You wouldn't see a pH of 11. Okay, it varies between 4.8 to 8.9, and that's about the limit. Okay, so D would be the right answer on that one. you wouldn't see a normal pH of 11. The part of the nephron that maintains the hypertonicity of the renal medulla is this is the part of the nephron that has the differential properties of in other words, the part of the nephron that has what we call differential properties like the nephron loop does. Remember, if you remember the nephron loop of Henle is permeable to water in the descending limb, but impermeable to water in the ascending limb. On the other hand, in the ascending limb, it is permeable to sodium. Okay. So what that does, it allows water to go into the descending limb and salt to go out of the ascending limb, and that creates an osmotic gradient. Okay. So the nephron loop is what maintains the hypertonicity, the high osmotic gradient. Okay. And so as you go into the medulla, as you descend deeper into the medulla, the more concentrated the urine in the interstitial fluid. That's because of those differential properties of your nephron loop, because of those unique differential properties. The descending and ascending limb of the nephron loop. Okay. So that's why you have a hypertonicity of the renal medulla. If you didn't have that, you would not be able to produce a concentrated urine. If this were not present, there would be no osmotic gradient to pull water out of the descending limb. Okay, that's why you want you don't want to avoid drugs like diuretics too much. Okay, because you want that nephron loop to be able to function correctly. Okay. When urine enters the collecting duct, it is blank to the blood. Well, remember, blood is isotonic, okay? It's about 280 to 300 milliosmoles. Okay? That's basically the plasma osmolarity. Plasma osmolarity, okay? But the urine can be as little as 50 milliosmoles. or as much as 1,200 milliosmoles. Okay. Very concentrated is 1,200. Very diluted, it would be 50. Okay. Remember the specific gravity of the urine varies between dilute, which would be 1.001, to very concentrated at 1.035. Okay. So 1.035 would equal about 1,200 milliosmoles. And 50 osmoles, 50 milliosmoles would equal 1.001, which is about, remember water is 1.000. Water has specific gravity equal to 1.000. Okay, so if it's 1.001, it's almost like water. 1.001 is almost like water. Okay. But so it would be what? Hypo or hyper? It'd be hypotonic. So it'd be hypotonic. So your urine would be hypotonic when it enters the collecting duct to the blood. Which part of the nephron employs the countercurrent mechanism? Glomerulus, nephron nucleus, or JG complex? Remember, the countercurrent mechanism is where? Nephron loop. Okay, remember nephron loop? Remember, nephron loop, that refers to the, it's a recta with blood going in opposite direction, and tubular fluid, That's the term current. All right, so I guess we'll stop there, and I'll let y'all, oh, it's already 12 o'clock. Y'all take care. I didn't realize it was 12 o'clock. I guess I ran a little bit over. All right, well, guys, well, good luck on the practical on Friday, and I guess this will be our last visit. Make sure you look at all those medical Jeopardy questions, and I'll keep you updated. Okay, if you have any questions, you can just email me. All right, y'all take care now. Good luck to you, and good luck on your finals.